In the vast world of synthetic polymers, few materials exhibit the versatility, functional breadth, and commercial significance that carbomers possess. Developed primarily as cross-linked polyacrylic acid polymers, carbomers are the backbone of countless formulations across pharmaceuticals, cosmetics, and industrial products. Their unique rheological properties, biocompatibility, and capacity to form highly viscous gels at low concentrations make them indispensable in product engineering, where controlled viscosity, suspension, and stability are paramount. In this video, we will dissect the structural chemistry of carbomers, examine their physico-chemical properties, elucidate the mechanisms underpinning their functional behavior, and explore their diverse applications across sectors. Carbomers are high molecular weight polymers derived from acrylic acid, systematically cross-linked with polyalkyl ethers or divinyl glycol. The polymerization process results in a three-dimensional, highly branched network structure, rich in carboxyl functionalities. These carboxylate groups are pivotal, as they confer ionizable properties, enabling the polymer to swell in aqueous environments and form viscous gels upon neutralization. The degree of cross-linking, molecular weight distribution, and residual monomers critically dictate the performance attributes such as viscosity, yield value, and spreadability. From a synthetic standpoint, the polymerization of acrylic acid via free radical mechanisms under controlled conditions ensures precise tailoring of the polymer's physico-chemical attributes. Modifications in the cross-linker type and concentration allow manufacturers to design carbomers with specific rheological behaviors suited for either shear thinning or highly stable gel formations. Importantly, the purity of the starting materials and stringent control of residual monomers are essential, particularly for pharmaceutical-grade carbomers, to meet regulatory requirements and biocompatibility standards. The remarkable utility of carbomers stems from their ability to undergo dramatic swelling in aqueous media, often expanding many times their dry volume. Upon neutralization with bases such as sodium hydroxide or triethanolamine, the ionization of carboxyl groups induces electrostatic repulsion within the polymer matrix, generating a highly expanded network. This process not only increases viscosity, but also imparts thixotropic or pseudoplastic flow characteristics, beneficial for ease of application and stability under static conditions. Furthermore, carbomers exhibit excellent suspending properties, making them ideal for formulations requiring homogeneous dispersion of insoluble particles. Their pH-sensitive behavior is another hallmark feature, allowing carbomers to be employed in controlled drug release systems where sol-gel transitions are triggered by physiological pH changes. Thermal stability, optical clarity in gel form, and compatibility with a wide array of active ingredients further consolidate their versatility. Carbomers are integral to numerous pharmaceutical preparations, particularly in topical, ophthalmic, and transdermal formulations. In topical gels, their capacity to provide a non-greasy, stable, and bioadhesive matrix enhances the efficacy and user compliance of active pharmaceutical ingredients, APIs. For ophthalmic applications, carbomers are valued for their mucoadhesive properties, prolonging the residence time of drugs on the ocular surface, a critical factor for therapeutic success. In cosmetics, carbomers serve as key thickeners, stabilizers, and emulsifiers. They enable the creation of elegant textures in creams, lotions, and serums, providing the desired aesthetic feel and functional stability. Notably, carbomer-based formulations can maintain product integrity across variable temperatures and storage conditions, safeguarding the activity of sensitive compounds such as vitamins and antioxidants. Beyond healthcare and personal care products, carbomers are employed in industrial settings as rheology modifiers in coatings, adhesives, and lubricants. Their ability to modulate viscosity without compromising the flow behavior makes them ideal for applications where precise control of material performance is required under mechanical stress or varying environmental conditions. Emerging research has also explored carbomer-based hydrogels for biomedical engineering applications, including wound dressings, tissue scaffolds, and drug delivery matrices. The tunability of carbomer networks allows for the incorporation of bioactive agents, providing a scaffold that not only supports cell proliferation, but also offers controlled therapeutic release. Innovations in nanotechnology have further expanded the potential of carbomers in the development of nanocomposite hydrogels for targeted therapies and responsive biomaterials. Carbomers, particularly pharmaceutical grades like carbopol, undergo rigorous quality control to ensure compliance with pharmacopoeial standards, including the United States Pharmacopoeia (USP) and European Pharmacopoeia (PH-EUR). Toxicological studies have consistently demonstrated the safety of carbomers when used as excipients, with low irritation potential and minimal systemic absorption. However, attention must be given to residual monomer content and the pH adjustments in final formulations to avoid adverse reactions. In conclusion, carbomers represent a cornerstone of polymer chemistry with profound implications across multiple industries. Their distinctive structural and functional properties have positioned them as a vital component in product innovation and therapeutic development. As research progresses, the evolution of carbomer-based materials promises even broader applications, fostering advancements in healthcare, personal care, and sustainable materials engineering. Thank you for joining us on this in-depth exploration of carbomers. For further insights into advanced materials and their transformative applications, stay connected with us and explore more scientific content.